today for your bell work, so if you, for your bell question, drew something that looked like this, and then you went ahead and divided it like so, and then you took this and then said 1, 2, 3, sort of kind of what you drew? Yeah. And what did you put right here? You can't see where I'm pointing. What did you put in this one?
G1.
And in this one?
S.
And here?
G2.
All these things added up we called
Interphase.
and most of the cells that you saw on Friday were in? If you interpret this diagram as a pie chart, why would most of the cells be in interphase when you looked at a sample of cells?
Because they're not all going to be like continuously growing. They have to like have a resting stage.
So every cell has to rest before division. Not all tissues need to divide. You said it's accurate too. And which phase takes the longest? So if you're looking at a random sample, most of them are going to be in interphase, right? Now when we see a cell in interphase, how do we know? How do we recognize that it's in an interphase? You can see a nucleus with a nucleolus, a clearly defined nuclear envelope. How many of you are comfortable with all of those vocabulary words so far? Okay. So when we're looking at a cell in interphase, we'll draw an animal cell. How do I know what I just drew is an animal cell, by the way?
Round.
It's round. And it doesn't have a cell wall.
I think everything. There's no vacuum.
So the question was, what's a nuclear envelope? Well, here's the nucleus. That whole thing, right, is the nucleus. The nucleolus would be that thing on the inside. The nuclear envelope is the membrane that defines the border of the nucleus. And in interphase, it's intact. So there is a completely intact nucleus. You aren't going to see anything else in a cell in interphase. Are there other things there? Absolutely. But when you look at them under a microscope, it just looks like a cell with a nucleus and nothing else. You will not ever see chromosomes. So you will never see chromosomes. During interphase. You will never see a chromosome during interphase. When I left you guys on Friday, one of the last things we were doing was actually looking at a diagram of a chromosome. And it was that little X-shaped thing, right? So you are never going to see condensed DNA. It's unfurled and being used. It's never going to be in the form of that X-shaped thing during interphase. This light turns on and off, brighter and darker. Sometimes, if I'm having a really perfect moment, I'll say something brilliant and it will get brighter. Those are very exciting moments. There are also very few and far between. After interphase, then we begin mitosis. Now here's where I'm worried about some of our vocabulary words. So raise your hand if you're unfamiliar or uncomfortable with the term as I list some for you. Chromosome. Mostly comfortable? Okay. Centromere. Sister chromatid. Spindle fiber. Okay. First thing we'll do before we enter mitosis then is tell you one of the first things that's going to happen as mitosis begins is that DNA is going to condense into chromosomes. So mitosis is the division of a nucleus. Mitosis is the division of a nucleus. And to make sure everything gets divided out evenly, the first thing that has to happen is that DNA will condense into a chromosome. So let's take a look at the anatomy of a chromosome. So you've often probably seen chromosomes drawn as an X-shaped thing. Each arm or leg is called a sister chromatid, or sometimes just a chromatid. The middle of it, where I drew that little white dot, that's called a centromere. What's on one leg of a chromosome is absolutely identical to the one that's next to it. So, this arm right here that I just colored in contains the exact same genes as the arm next to it. So, for example, some things, let's just pick a silly one. Give it a try. Don't worry, it is not a fault, flaw, or defect. It's a gene. Your ability to is inherited. It's true. Of all the random things you can inherit from your parents, if you can't do it, you will never be able to do it.
I'm going to try for the rest of my life.
You know what's actually sad? I love my mother dearly. My mother is a phenomenal woman. God bless her soul. We used to tease her because she can't do it when we were kids. We were always like, come on, Mom, just try. Just try. And she would sit there for I don't even know how long, like trying to force herself to do it. And we were always like, Ma, you know it's never going to work. Like, that's why we do this to you. Like, you're never going to be able to do it. But poor Gail, she's just one of those people who's so determined, like, I'm going to try. I know I can't. No, really. You're never going to do it. So if that gene, this gene, were located in that yellow portion of that chromosome, the exact same gene is going to be on the arm next to it. Think of it this way. It's like a photocopy. But to make sure that our photocopy stays together, we attach them in the middle. Right, Carmen? Excellent. Why do we do it?
Because it's in our genes.
Stays together. Okay, so I know there's a centromere in the middle and there's like divided in half. Are they all exactly the same, all four?
No. So basically, what's on the top is different than what's on the bottom here, but they're identical to the one next to it.
So the sister chromatids are the same?
Yes. The sister chromatids are identical. Now, the reason that we have to make sure we understand that is because it's going to become very, very important for us in anaphase. So as we proceed through mitosis, that fact will become very important. Look at it this way. If I were to make a photocopy of a series of pages for you, say I had a section of a textbook that I thought was a great textbook and I wanted you to have a copy of it. It would become essentially useless to you if I accidentally left out some pages. So what we do is make sure that we have an identical copy of those pages, and then we pull them apart. Okay, I say that like teachers do that for you. It's not true. We have the photocopier do that for you. But they get pulled apart so you have an identical, complete set. We attach them at the center to make sure that when the new nucleus is made, there's an identical set of DNA in both nuclei.
So the ones that are identical, are they the ones that are next to each other, or like
Mhm.
above and below?
Next to each other. So they're identical. They're in the nucleus. So they're identical. This one, I don't know how to indicate the one that I refer to. So that one is identical to this one, and this one is identical to that one. So they're identical.
But they're not identical to each other.
Right. But top and bottom are not identical.
Isn't the whole thing called the sister chromatid?
This,
Just the leg and one they're called.
this is one sister chromatid. This is the other sister chromatid. And the reason we call them sisters is because they're identical. That's to indicate that they're the same.
See how it's like, you circled them like this?
Hmm.
What if they were circled like that? Would those two be like, I don't know.
So,
Like this, like the two, like the top one?
You said the top ones were
The
identical.
two bottom ones are
Like, so like, one sec. So you know how you circled, so you know how you circled like those?
those two, see how they're labeled as one? They're the part of the same sister chromatid, but they don't have the same genes. So tongue rolling would be on the top and hair color would be on the bottom.
So like, the two bottom ones aren't sister chromatid?
This whole big thing is a sister chromatid. So the whole thing up and down, that's one sister chromatid.
Just up and down, no?
And this is its identical copy.
So the identical ones are not sister chromatid.
This is the sister to that one.
Yeah.
Mhm.
But like,
They're two different sisters.
so one and one are the sisters.
One and one. So here's what I think, and this is an issue of my drawing. This piece right here is solid. It's not four separate things, it's two long things. Light bulb. Two long things joined in the center. This may help. If they are. Very long. The School of Education would force me at this point to apologize to you for playing into gender stereotypes, where pink represents girl and blue represents boy. Sorry.
Real men wear pink.
Real men wear pink.
Sister chromatid, sister chromatid, they're sisters to each other. They're joined in the center, where they are, in fact, this top half contains all the same DNA as this top half. This bottom half contains all the same DNA as this bottom half. There's only two things here, this one and this. They're not four separate pieces. Does that make sense?
Yeah, but which ones are like the sister chromatid?
These two are sisters to each other.
So up and down are sisters. Okay.
No, this one is identical to this one.
No, the bottom, yeah. The top and bottom.
Yes. The reason that we typically refer to these as arms and legs is because you understand that arms and legs are different from each other, right? So arm and leg,
Arm goes to arm, leg goes to arm.
yes. So that's helpful. Did everybody hear that? The reason on sister chromatids we refer to it as arm and leg is because the arm is the same as the arm, and the leg is the same as the leg.
Oh, and then they're different. Everyone's having
Yes.
is having a
Yeah, we're all with me now? Excellent.
But because they're on the same side is why they're like.
Yes. Exactly. Better? Okay. Usually, I used my thumbs as the centromere, but in real life, they're actually joined by a little piece of protein in the middle. That's the centromere that holds them together. So when we're going through those vocab words that we were like, I'm not entirely totally sure I'm okay with, centromere is the thing in the middle. This structure does not become visible until prophase. So when prophase begins, DNA condenses into a chromosome. That's how we're going to know that we've entered prophase. Something else happens in prophase. You remember what it was from all of your... Ooh, spindle fibers. Do you guys remember centrioles from your organelles? They're little star-shaped things, little churro-shaped things, right? They turn in pairs at right angles to each other. And it is likely your first semester teacher just said simply this. They are responsible for organization of cell division, and they moved on.
That's pretty much.
They're only in animals.
And they are only in animals.
Yep.
Yep, right in the middle.
Teachers, please excuse the interruptions.
Sure.
Just a reminder that in a few minutes, you will be getting report cards for your fifth hour students. Just ask that you hand those out by the end of the hour. Thank you.
Okay,
What time do we leave this class?
it's 1:10.
1:10.
I don't leave this room until 2:30, so then I'm like, 2:30?
Oh, okay.
Join honors bio next hour, and then the hour after. So, this structure becomes visible. Spindle fibers form from the centrioles. So you understand from all of the work that you've done so far that these are somehow going to separate and move to opposite sides of the cell, right? Think of a centriole as like a fishing reel and a spindle fiber as the fishing line. It's going to attach to the center of this, and then the spindle fiber is going to pull to the opposite sides of the cell.
What happens to
The centromere is going to dissolve, but it won't do that for another couple phases. This part, prophase, is where the centriole, hooks onto the centromere. So it's easy to remember in that if we think about it, centriole and centromere get attached. They begin with the same letters. So centriole attaches to centromere through a spindle fiber.
So the centriole. The centriole is on.
It actually releases it. Mhm. So prophase we can see a chromosome now, right? Recapping. Spindle fibers attach from the centriole to the centromere. So it goes from the centriole to the centromere. And for those of you who asked what the nuclear envelope was, well, here's why we had to learn. The nuclear envelope begins to dissolve during prophase. The nuclear envelope begins to dissolve during prophase. So if we draw our cell again, we have a circle. And our nuclear envelope is typically then drawn here as a dashed line to indicate it's dissolving. And inside that dashed line, we have chromosomes because they're visible now. And if I had drawn my cell bigger, we could draw some spindle fibers in there, but I made it tiny, so. If you have that worksheet out in front of you, knowing what we just said, did you label cell 1, 2, 3, 4, or 5 as prophase? That's quite all right. I don't mind. After picture number 4.
Were the spindle fibers the long
Yep.
sides? Okay.
And then those are supposed to be where the centrioles are.
3.
Picture number 3. What phase comes after prophase? So we go prophase, metaphase, PMAT. Prophase, metaphase. Metaphase is actually one of the simplest ones to recognize because all of the chromosomes are aligned at the center of the cell. So if you see a cell where all of the chromosomes are all lined up in a straight line, that's always going to be metaphase. That particular region of the cell has a couple of different names we can call it. We can either call it the equator, which you guys prefer because, well, you have a natural understanding of what an equator is. You could also call it a metaphase plate. Students love the question, in what phase do chromosomes align at the metaphase plate? Because you better get the answer right. They align at the metaphase plate during metaphase. You'll notice also that centrioles are now on the opposite poles of the cell. So this region is called a pole. That's called an equator. Are there any other conditions in which you have taken a sphere, divided it with an equator, and indicated that there are poles on either side?
The Earth,
Yeah.
planet Earth.
If we take a look at a globe. So we do the same thing here. Say equator, poles. During metaphase, centrioles are at opposite poles. And of course, we still have spindle fibers attached. So when you guys play tug of war, there's like that during faculty student,